Welcome back everyone to Equity Tutors. The module that we're currently working on, if you've been following the lessons, is homeostasis. And Lauren last week gave a good overview of what homeostasis means. And today we're going to learn about our first very specific example of homeostasis in the body. So homeostasis just means that we are keeping the environment of the body regulated the same. And the example that we're going to look at today is water. So how do we keep the concentration of our water in the body to exactly what it needs to be? Because consider that our bodies endure lots of different situations, changes in heat, for example. So you may sweat more one day, sweat less another day, drink more one day, drink less another day. You may drink alcohol that makes you lose more urine. You may be taking more medications that make you lose more urine. So we're going to talk about how our body, throughout the different changes in the environment and while we're eating and drinking, can maintain the same concentration or content of water. And the organ that is responsible for regulating this are the kidneys. So we're just going to start firstly in this lesson with going over the anatomy of the kidneys and the kidney structure, and then delve further into each of those components, and then finally talk about the kidneys as a filtration system, how that works, and or how the kidneys can compensate for different changes in the environment or in the body that would change the water concentration. So the kidneys are a pair of red-brown organs attached to the back of the abdominal cavity. So they're at your lower back, and they are surrounded by a thick protective layer of fat and connective tissue. If you place your hands on your hips, your thumbs are basically where your kidneys are, or they're sat behind your thumbs. And the kidneys are supplied with blood from arterial pressure, so the blood comes directly from the heart to the kidneys via the renal arteries, which is a branch of the abdominal aorta. And then blood leaves the kidneys through the renal vein into the inferior vena cava. So you can see down this very simple diagram here, showing the aorta coming from the heart, going into the kidneys, and then the blood vessels coming out of the kidneys and going back to the heart via the inferior vena cava. And the kidneys are important in blood filtration, excretion, and osmoregulation. And osmoregulation just means keeping the water concentration of the body steady. So it's a type of homeostasis. So delving deeper into the kidney structure. So the kidneys are made up of millions of these tiny filtering units that we call a nephron. So there's a couple of different diagrams that you need to learn and label here. So firstly, you need to be able to label the sort of macroscopic view. So that means the bigger picture of the kidney. So the kidney, as you can imagine, seeing yourself with your own eyes, and what the organ actually looks like. And then when you look more closer up, so microscopically, that's where we can start to see the structure of the nephron. But you also need to understand how the nephron, so these filtration units, are organized within the larger kidney image. So starting with the cortex. So the cortex is this dark outer layer. This has a high density of capillaries, and it is the site of blood filtration. So the cortex makes up the Bowman's capsule, which we're going to learn about shortly, the convoluted tubules, and the blood vessels. Don't worry about these words too much yet. We're going to get to them at the end of the lessons, but just know that's where these structures are located. Then we have the medulla. So this is a lighter area which is inside of the cortex, and this is where the nephron tubules are located. And you can see in this image, you can see that there is between the cortex and the medulla, you can see a source of both arteries and veins separating the two. We then have the pelvis. So the pelvis is the innermost part of the kidney. This is actually what is going to collect the urine before it passes down to the bladder. Then we have the ureter. This is the tube which connects the kidneys to the bladder. So after the pelvis and that tube is continuing down, we call that ureter. not to be confused with urethra, which is the tube that comes from the kidneys. And then you may also get an image with the bladder labelled on there too. So that's going to be the muscular sac, which actually holds the urine. So let's just quickly recap that. The four main terms I want you to take away from this particular diagram. You have the cortex, which is the outer layer, the medulla, which is the inner layer, and then the pelvis, the innermost layer, which leads to the ureter, which goes to the bladder. Now I want to delve in more to the microscopic structure of the kidney. And I mentioned that in the cortex is where we have something called the Bowman's capsule, and in the medulla we have the biggest part of the nephron, which are these small filtration units. So now if we move on to the next slide and look at the structure of the nephron, I have labelled here cortex and medulla, so you can see what I mean. So this is how the nephron is organised within the kidney itself, and you can see clearly which parts of the nephron are positioned in the cortex, which parts are positioned in the medulla, and they are organised in this way all along the outer rings of the kidneys, facing inwards towards the pelvis. So try and picture that more clearly. So the nephron, as I said, is where blood is filtered. So essentially what we're going to do is take everything that we need from the blood and get rid of everything that we don't need via the urine. So the nephron is where blood is filtered, and the majority of everything that's filtered will actually be returned to the blood. It's very minimal things that actually get excreted in the urine. So it removes things like nitrogenous waste, balances mineral ions, and also water levels. So not only is it balancing water levels, which I mentioned at the beginning of the talk, but you could also have, for example, an abundance of a certain mineral, and when that exceeds what the body needs, and in some cases can become toxic to the body if we have too much of a certain mineral, that will be excreted. So each nephron is around 3 centimeters long, and they're around 1.5 million per kidney. So this provides the body with several kilometers or worth of nephrons to allow for this reabsorption of water, glucose and salts after the blood has been filtered. So when you look specifically at this diagram of the nephron, let's just go through a couple of the basic structures. I want you to first notice that what we have is two different systems intertwining. So we have the blood flow, as you can see by the red and blue lines, but in between now we have this series of tubes, and this is where the urine is going to be collected, but there's going to be lots of exchange between the blood flow and this tubing, which is ultimately going to lead to the bladder and where the urine's collected. And there's going to be lots of exchange. There's going to be some basic filtration, there's going to be some osmotic pressure, and we're going to talk through each of those steps one at a time. But let's just go through the structure to start with. So let's just start with the Bowman's capsule and the glomerulus. So the Bowman's capsule is highlighted here in a square in the image. And what you can see is it's acting like a cap. And within that cap, you can see a tangle of capillaries sitting within that capsule, and this is where ultrafiltration is going to take place. So this is where all the blood is coming in under really high pressure and then essentially gets filtered out into the Bowman's capsule, which is leading through this series of tubes. So the name of this tangle of capillaries is called the glomerulus. So all the pressure coming from generated by the heart through the arteries forces all solutes in the blood plasma to be forced through the capillary walls here. We're going to go through this in more detail in a second, but just to give you an overview, this is what's happening. Imagine lots of pressure in these capillaries pushing everything out into the Bowman's capsule. From the Bowman's capsule, we then go into the proximal convoluted tubule. So this is the first coiled region of the tubule, and this is where products needed in the blood So things that have just been filtered out of the blood are immediately going to be reabsorbed back into the blood. So look again at the structure in this image. The arteries are still tangled around this proximal convoluted tubule. We then have the loop of Henle. So this is this really long structure that's it's going to descend and then ascend back into the cortex. So the loop of Henle is all in the medulla. And the loop of Henle is a long loop tubule which spans the cortex and the medulla, so all the way into the medulla. And this is going to be used to concentrate the urine. So a salty environment is created in the medulla in order to cause water to come out of the tubule. So we maintain a salty environment in the medulla, which makes the water that was filtered through the glomerulus into the Bowman's capsule, through the proximal convoluted tubule, and then into the loop of Henle to move out. Again, we're going to go through this in much more detail. This is just very much an overview. And then from the descending limb, we have the ascending limb. And in the ascending limb, we still have movement of water and other things in and out. And then finally, number 8, your label in the diagram, we have the distal convoluted tubule. And then this is the second coiled region of the tubule. This is present in the cortex, where osmosis and diffusion of solids occurs in order to just fine-tune the water potential and the pH of the blood. So this is the final stage where we can finally tweak the different components in the urine, maybe get back things that we may need more of before it's finally lost into the urine. And then the final part of this diagram, or the final structure of the nephron that you need to know, is the collecting duct. And this is where the urine travels through the collecting duct down to the pelvis of the kidney and then through the ureter to the bladder. So I'll just go over that one more time. Starting from the top, and this black square indicates on the top left of the image of the nephron. So this is the Bowman's capsule, and the blood is essentially being filtered into the Bowman's capsule via the glomerulus, and the glomerulus is receiving blood through the afferent and efferent arterioles. So that's just top and bottom, efferent and afferent. They feed into the glomerulus, make a really high pressure environment, and that is filtering all sorts of things out from the glomerulus into the Bowman's capsule. We're going to go over that in more detail at the end of this lesson. From the Bowman's capsule, we then have a coiled region of the nephron called the proximal convoluted tubule. And again, note that throughout all of this tubing present in the nephron is intertwined with a blood supply. So from the proximal convoluted tubule, we then go into the loop of Henle. That is split into the descending limb and the ascending limb. They are both present in the medulla of the kidney. Out from the ascending limb, we then have the distal convoluted tubule. Again, this is another coiled region, and this is the final part of the nephron where there is fine-tuning of the contents of the urine in respect to what the body needs in terms of sugar, salts, minerals, and water content. And then the final part, final structure of the nephron that you need to know is the collecting duct. And that is going to lead through the pelvis of the kidney to the ureter to the bladder. And you can see all of those structures in the second image here too on the right-hand side. And again, I just want to emphasize how the tubules and the loop of Henle really intertwine with the blood supply to allow for constant exchange, essentially, of the contents of the urine with the blood. Okay, so how do the kidneys actually process water and these salts, sugars, minerals to keep them at a regulated concentration in the blood? So this is a 4-step process. Today we're probably just going to cover step 1, which is the ultrafiltration, and then the remaining 3 steps will be covered in the next lesson, or part 2 of this lesson. And before we start talking about ultrafiltration, I just want to remind everyone of some key terms from past lessons that you're going to need to know and remind yourself of to completely understand today's lesson. So the first one is osmolarity. So this is just referring to the concentration of a solution expressed as a total number of solid particles per litre. It just means the concentration. So we're just saying, for example, how much salt is in the water. So to repeat the definition of osmolarity in the context of salt, we would say concentration of salt expressed as a total number of particles per litre of water. So that's grams per litre, for example. The next term is isoosmotic. So this is to have the same osmolarity as another fluid, meaning the osmotic pressure would be the same. There would be no movement of water either way. Equilibrate. This means to bring to the state of equilibrium, which means that it is staying in the same state. So even if there is movement either way, ultimately the net outcome is the same. And then interstitial fluid. So this is the fluid which surrounds tissue cells. So as I mentioned a second ago when I was talking about the medulla, the tissue cells within the medulla or the interstitial fluid in the medulla can also itself maintain a certain osmolarity. And that is going to help in this filtration process. So we're now going to start diving into ultrafiltration. So the blood enters the kidney via the renal artery, which is under pressure from the heart, and then into a complex network of capillaries called the glomerulus. So that circular structure of capillaries that sit inside the Bowman's capsule. Water and all soluble plasma components are then forced out of the glomerulus. So think of it as like a filter. It's like a sieve. Anything small enough to pass through the walls of the arteries, so things like sugar and water, are going to be pushed out of the capillaries into the Bowman's capsule. However, large proteins are too big to fit. And this pressure created in the glomerulus to allow for this filtration to happen is generated by the differences between the afferent and efferent arteriole. So remember I mentioned in the previous diagram you have the efferent arteriole at the top and the afferent arteriole at the bottom, and now where you see this closer picture of the glomerulus, what you can see is the afferent arteriole is actually bringing the blood in to the glomerulus, and the diameter of that afferent arteriole is wider than the efferent arteriole, and that helps create this pressure within the glomerulus to force these solutes out of the glomerulus and into the Bowman's capsule. So let's go through that one more time with some added details. So the arterioles branch off the renal artery and lead to each nephron. They form a knot of capillaries called the glomerulus, and they sit inside the cup-shaped Bowman's capsule. The capillaries get narrower as they further divide into the glomerulus. They get smaller and smaller, and this increases the pressure on the blood moving through them as they run out of space to flow, essentially. And this eventually causes the smaller molecules being carried in the blood to be forced out of the capillaries into the Bowman's capsule, where they will form what is known as the filtrate. So anything that's come out of the blood into the Bowman's capsule is called the filtrate. The blood in the glomerular capillaries is separated from the lumen of the Bowman's capsule by two cell layers with a basement membrane between them. And you can see this again in this diagram at the junction between the Bowman's capsule and the glomerulus itself. So this first cell layer is the endothelium of the capillary. So each capillary, every capillary in the body is lined with endothelial cells, and they are perforated by thousands of tiny membrane-lined circular holes. So they always have a certain level of permeability. The next layer is the basement membrane. So this is made up of a network of collagen and glycoproteins. This is tougher to get through. And then the second layer of cells, at least, so we have three layers total. Two of them are layers of cells. One is the basement membrane. The second cell layer is the epithelium of the Bowman's capsule. So we have glomerulus, which has a cell layer, then we have the basement membrane, and then we have the Bowman's capsule cell layer. And the second layer, the Bowman's capsule cell layer are epithelial cells, and these have many tiny sort of projections with gaps in between them, and these are called podocytes. So this is this irregular shape that you see on the diagram. It's not a smooth line like you see for the cell layer of the glomerulus. It's more bumpy, and these are called the podocytes. And these are important because otherwise epithelial cell layers would not allow for the solutes exiting the capillaries to be absorbed into the Bowman's capsule. So they are specially adapted cell types on the Bowman's capsule to allow for the filtrate to come through into the Bowman's capsule. And as I said, the fluid that filters through from the blood into the Bowman's capsule is known as the glomerular filtrate. And the main substances that are going to pass out from the capillaries to the glomerular filtrate are things like amino acids, water, of course, glucose, urea, and inorganic ions, things like sodium, potassium, and chlorine. Red and white blood cells will remain in the blood because they are too large to pass through the holes in the capillary endothelial cells. And the basement membrane's also acting as a filter here. So it's going to stop really large protein molecules from getting through. And so just to end the lesson, I want to talk about different factors that can affect the water potential in the glomerulus and how that affects the movement of water. So ultrafiltration occurs due to the differences in water potential between the plasma in the capillaries and the filtrate in the Bowman's capsule. So remember, water moves down a water potential gradient. So from a region of high water potential to a region of low water potential. So as the water potential in both the Bowman's capsules, or more likely in the glomerulus, changes due to environmental factors or other things that your body might be encountering, the amount of ultrafiltration that occurs is going to be different. So the first factor that can affect water potential is pressure. And this is all things that are going to affect your blood pressure, which is why people with high blood pressure, for example, commonly have kidney problems. So how does pressure affect water potential in the glomerulus? So as the afferent arteriole is wider than the efferent arteriole, the blood pressure is relatively high in the glomerular capillaries. So this then is going to raise the water potential of the blood plasma in the glomerular capillaries above the water potential of the filtrate in the Bowman's capsule. And what happens when you raise the water potential? Water will move down the water potential gradient from the blood plasma in the glomerular capillaries into the Bowman's capsule. So higher blood pressure, more water being moved into Bowman's capsule. And then the second factor I want to discuss is solute concentration. So things like sugar and salts. So whilst the basement membrane allows more solutes within the blood plasma to filter into the Bowman's capsule anyway, and plasma protein molecules are too big, they stay in the blood. But as a result of this, the solute concentration in the blood plasma in the glomerular capillaries is still higher than that in the filtrate of the Bowman's capsule, because you're going to get a concentration of protein in the capillaries. So even though you're losing all the solutes, you're still getting an accumulation of the proteins. So then what this means is the water potential in the capillaries is actually lower than the filtrate in the Bowman's capsule. And so what happens to the movement of water in that case? Water will then move down the water potential gradient from the Bowman's capsule into the blood plasma. So what I'm trying to illustrate here is two things. Firstly, to demonstrate how changes in the pressure in the body and solute concentration in the body will affect how ultrafiltration occurs, but to also demonstrate to you that this is a balance. So these two factors are actually working against each other. The increased pressure is going to make water move out, but the solid concentration difference is going to make water move into the glomerulus. So it's a two-way thing. But overall, generally, the effect of the pressure gradient is going to outweigh the effect of the solid gradient. So what that means is the net movement is still going to be from the capillaries into the Bowman's capsule. Therefore, the water potential of the blood plasma in the glomerulus is higher than the water potential of the filtrate in the Bowman's capsule. This means that as blood flows through the glomerulus, there's overall movement of water down the water potential gradient from the blood into the Bowman's capsule. And we can see that in this diagram here. So looking on the left-hand side, this is where the glomerulus is. And then you can see in purple plasma proteins, which are too large to pass through the basement membrane, which you can see as this layer with the squiggly lines in. And then what happens is that you get water movement and solid movement from that capillary across the basement membrane into the region of lower water potential, which is the Bowman's capsule on the right. So you get movement of water, but you're also getting movement of solids back into the capillaries because of this concentration of proteins in the capillaries on the left-hand side. So that's that smaller arrow of movement of water due to solid concentration gradient, but ultimately this movement is still less than the movement due to the pressure that you get. And so overall, net impact is accumulation of amino acids, water, glucose, urea, and inorganic ions present in the filtrate, which is the glomerular filtrate in the lumen of the capsule. So I'm going to leave our lesson there for today. So we covered what are the kidneys actually regulating, and how that relates to homeostasis. We went over the structure of the kidney, and then we went over the first step in how the kidneys work, which is ultrafiltration. And what we'll do next week is to go over the remaining 3 steps, which we will refer to as selective reabsorption, firstly in the proximal tubule, then in the loop of Henle, and then finally in the distal convoluted tubule and the collection duct. So thank you so much for listening. Remember, you can access additional content on our Patreon page by searching for Equity Tutors, where we have a second 30-minute lesson every week, plus monthly bonus content. You can also find us on most social media platforms. We will keep you updated on new content, and you can find us there by searching for Equity Tutors UK. Please like, share, subscribe and comment wherever you are listening, and if you're enjoying, please leave a review. Bye!
Bye!